So I'm going to give you the four-stage hypothesis that gives us the origin of life. I'm going to give this to you in four stages, and this sounds like a very nice short response question I can ask you guys. Stage one, synthesis of organic compounds. So I need to make something that's inorganic, those volcanic gases I just talked to you guys about, into something organic. So again, just to reiterate those gases, we've got water, methane, ammonia. I can see that here in this experiment. And those are not friendly to life, and they're not life in itself. These are inorganic molecules. Life depends on more complex organic molecules, like I taught you guys in the macromolecules chapters. We've got sugars, fatty acids, amino acids, nucleotides. All these guys are composed of the same elements that those inorganic molecules are composed of. So how do we go from inorganic to organic? How easy is that? Is that even something that can happen? In 1953, so mid-20th century, we actually have this really cool experiment that showed us that this could happen. It's actually quite an easy experiment to replicate, to show us that you can go from inorganic to organic, given a couple of conditions. So basically you have an apparatus, and in this apparatus you have, to emulate volcanism, you have sparks emitting lightning as well, in addition to the gases that are produced. And so you actually have some energy in that setup. And so then what happens, so this is an atmosphere, quote unquote, that you're creating of early Earth. Give it enough time. Actually, they ran this experiment for about a week. We have condensation occurring. So then we actually have a sea here. So that's going to emulate an early ocean, very high temperatures. And then we have gases being formed. So those gases then recirculate and go through this early atmosphere again, and condensation, samples collected. It's now this sample, after it's run through that apparatus, that you can collect. And those scientists determined that through that sample, they actually found a bunch of organic molecules that were essential for life. They actually found the monomers that would then make amino acids, proteins, had collected in the sea. So that was pretty neat that you could go from something inorganic to organic given these conditions that were thought to exist in early Earth. Scientists are also considering other hypotheses for this inorganic to organic compound. This is the widely accepted one. The other ones that I do want to include, that I do want to show you guys, is that we could have life originating in volcanoes themselves, deep sea hydrothermal vents, those very high temperature areas in the sea that I'm actually going to discuss, because we do see some really neat bacteria that like to live there. So this could have occurred given those high energy environments. Meteorites bringing along life is another hypothesis. Then coming to Earth, bringing those first organic molecules, not really needing to go from that inorganic to organic. Okay, so the first stage, inorganic to organic. We have organic compounds. This apparatus has shown me how I could do that, given Earth's early atmosphere. Stage two, now, in order to constitute life, we actually have to synthesize those organic compounds into something more complicated. I taught this to you. We need to go from those simple monomers to those more complicated polymers. We need to get those larger structures, amino acids. We need to get proteins. We need to get nucleic acids, DNA, RNA. We need to get those complex sugars to form and so forth. So early life, or before life, because we're talking about before life, we didn't have enzymes to help us with this. We have enzymes now to help us with this process. We didn't have that. Enzymes themselves are proteins, so you can't have a protein to start and make a protein, the very first protein to exist. So how did this occur? Well, scientists have found it does take a lot of time if you don't have an enzyme, but scientists have found that you can actually go from monomer to polymer by just dripping solutions of these monomers onto hot sand, clay, or rock. So if you have something that's a hot temperature, that sand, clay, rock, like early Earth would have had, you can actually, over much time, get a monomer to a more complicated polymer. Those bonds will just form themselves, given that high heat environment. Pretty cool. Stage 3. All right, now we got all these polymers. We've got those more complicated macromolecules. How do we contain them? We're talking about life here, and life has to have a container, if you will. I'm talking about the cell. The cell contains life and helps perform life's functions. So in stage 3, you're forming a pre-cell. So it's not a cell yet. Just think of it as a container to hold all these polymers. So really, this happens due to something called polarity. I taught to you guys about lipids and lipids being scared of water. And when you actually immerse lipids in water, it doesn't dissolve. It in fact forms these missile-like structures. Soap does this beautifully. When we see soap bubbles, they're actually little missiles that are forming because they're hydrophobic. They're afraid of water. And so this is a nice, it looks like a phospholipid bilayer, a cell membrane. This is actually not a cell membrane yet, but it's the beginning stages of a cell membrane. We can call this a pre-cell, and it's a really cool way to house these polymers that form stage 3 in this hypothesis. Stage 4, last stage for the origin of life. Okay, we have this pre-cell to house these polymers that are going to perform life's functions. We've got everything going. But life leaves offspring, whether that's in a clone of itself or sexual reproduction. And we're talking about an early life form here, so it's probably a clone of itself that it's making. So we need something to self-replicate itself in order to make a clone of itself, in order to continue on life, so that that 4-stage occurrence doesn't need to happen again, because it takes a lot of time and a lot of randomness for that to happen, and kind of have to get lucky for all those things to match up before we get to stage 4. So we actually see self-replicating molecules occurring here. This is inheritance that I'm talking to you guys about. So really, scientists have discovered a very simple way for life to replicate itself, in the fact that we have we actually have this, humans, it's probably a leftover from early life, that we have short strands of RNA that can replicate themselves quite efficiently. They don't really need any other beautiful proteins to help them with this self-replication process. These are called ribozymes that we have. They're actually in themselves enzymes that are nucleic acids that can then leave instructions for the next life form to exist. So that's stage 4, the origin of self-replicating molecules. Very early stages of inheritance. These are ribozymes. Natural selection, years later, would begin to shape properties of pre-cells that would have these self-replicating RNA for them to eventually become more elegant over time, giving us that double-membraned cell that we have, multicellular human beings, plants, diversification of species, adaptations to then give us more complicated structures. At some point, more stable double-stranded DNA would have come about, replacing that RNA to store genetic information, giving us the blueprint of life. Okay, the 4-stage hypothesis given to you guys in short image form. Inorganic compounds become organic monomers. This is the abiotic synthesis. That's stage 1. Stage 2, forming these monomers into these more complicated polymers. We now have larger structures. Now we have to house these polymers in something. We have to contain it in order to get life. Now we have membrane enclosed compartments. These are the formation of pre-cells, stage 3. Stage 4, we want to go ahead and replicate these instructions in order to continue on life as we know it. Stage 4, these are ribozymes, those very simple RNA structures that can copy themselves.